Well, hello, my fellow pre-meds, and welcome back to another video. In this video, we're going to be covering a topic that I think most pre-meds dread while preparing for the MCAT, and that is the topic of carbohydrate metabolism. I recognize that this is a difficult topic, a complex one, maybe even a painful one for some of you. And therefore, I've taken certain steps to hopefully make this process as, I don't want to say as easy, but at least as painless as possible for you all. The first step I've taken is that I really try to highlight and explain the underlying mechanisms behind the concept so that you don't have to rely too much on memorization. The next step is that I really limit the amount of information that you do have to memorize, because of course there are certain concepts you have to memorize. So I try to limit it to important enzymes, intermediates, steps, and pathways that actually show up on the MCAT. Also, you can use our Anki cards to aid with memorization. And finally, I take this broad topic of carbohydrate metabolism and break it down into three broad categories, into three broad slide decks to hopefully make it more digestible. In this video, we're going to be focusing on glycolysis, pyruvate dehydrogenase, glycogenesis, glycogenolysis, and gluconeogenesis. Don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow, and I would really appreciate it. So without further ado, let's get started. This is a diagram that shows you the relationships and the connections between the different pathways that are involved in this video. You can pause the video and think about them, but we're going to jump right into glycolysis. So glycolysis is the process of breaking down glucose. Glucose, lysis, breaking down. So breaking down glucose into what? Into 2,3 carbon molecules. So glucose is 6 carbon molecule, 2,3 carbon molecules, which are 2 pyruvate molecules. In the process, you get 2 NADHs and 2 ATPs. Or to be more accurate, you net 2 ATPs. And that's because we have an investment phase, an energy investment phase, and an energy payoff phase. The energy investment phase is where you invest 2 ATPs, you deposit 2 ATPs. The energy payoff phase is where you collect that paycheck with 4 ATPs, so you net 2 ATPs. This just shows the important reactants and products in the process of glycolysis. Let us get started with discussing each of the steps of glycolysis, and we're going to run through them because they're not all high yield, and the ones that are high yield, we will see them again in subsequent slides, and we'll really hammer them down. So starting off with glucose, glucose will be first phosphorylated using hexokinase, an irreversible enzyme, which we will see again because it is high yield. In this process, you consume 1 ATP, so that's that first investment. Next, you want to convert glucose 6-phosphate into its constitutional isomer using isomerase, so you get fructose 6-phosphate. Next, you want to phosphorylate fructose 6-phosphate at its 1' carbon to get fructose 1,6-bisphosphate. So that's fructose with 2 phosphates. This is a very important enzyme because it's a rate-limiting step. Again, you consume another ATP over here. Next, you want to break fructose 1,6-bisphosphate into 2 separate 3-carbon molecules, specifically glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now notice that there's an arrow between dihydroxyacetone phosphate and glyceraldehyde 3-phosphate because they are interconvertible. So essentially you get 2,3 glyceraldehyde 3-phosphates that can participate in the remainder of these steps. So all of these steps are going to happen twice in the energy payoff phase. The very first step here is to convert glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, a very important step. So we'll see it again in subsequent slides, but essentially you are generating NADH in this step. And you are also phosphorylating glyceraldehyde 3-phosphate into a molecule that now contains 2 phosphates, but you're not consuming ATP because you're using inorganic phosphate, so you're not wasting energy now. The next step, you are generating ATP by dephosphorylating 1,3-bisphosphoglycerate into 3-phosphoglycerate. So this is using 3-phosphoglycerate kinase. So we'll see this again because it's an important step. Next, we convert to 2-phosphoglycerate. Not very important. After that, you convert it into phosphoenolpyruvate, which is the second to last step. And then you convert that using pyruvate kinase into pyruvate, again, generating another ATP. So because this runs twice, you get 4 ATPs total and 2 NADHs. All right, now let's talk about the high-yield enzyme. So the first enzyme I wanted to talk about is hexokinase or glucokinase, which traps glucose into the cells. So as you'll recall from our video on biological membranes, glucose, because it's polar, cannot cross the plasma membrane. It needs this fancy membrane protein to allow it to transfer. Now, because once you phosphorylate it, you add another charged and polar molecule onto it, it can't even cross this membrane protein, and therefore it is trapped in the cell, and that is the purpose of hexokinase, which is an irreversible enzyme. Next, we have PFK1, phosphofructokinase, which is the rate-limiting step where it takes fructose 6-phosphate and phosphorylates it to produce fructose 1,6-bisphosphate. So that is a fructose molecule with 2 phosphates, one at the 1' and the other at the 6' position. Now, it is activated by AMP, and that makes sense. If you have a lot of AMP in the cell, that means you have little energy and you want to produce more, so ramp up glycolysis. It is inhibited by ATP and citrate because ATP, if it's in abundance in the cell, you do not need glycolysis to run, and citrate is an intermediate of the Krebs cycle. So if you have a lot of intermediates of the Krebs cycle, then that probably means you don't need glycolysis to run. Now, PFK2 is not directly involved in glycolysis, but it activates glycolysis. This is how. So fructose 6-phosphate can take one of 2 routes. It can either go the PFK1 route, continuing glycolysis, or be phosphorylated at its 2' position to get fructose 2,6-bisphosphate using PFK2. This turns out to be activated by insulin. So once you activate it, you get more F26BP to stimulate PFK1. So you want to think of it in terms of fructose 2,6-bisphosphate activates glycolysis, and as we'll see in future slides, it inhibits gluconeogenesis. The next enzyme I wanted to talk about is glyceraldehyde 3-phosphate dehydrogenase. So recall that is the first enzyme in the payoff phase over here. So it is phosphorylating glyceraldehyde 3-phosphate using inorganic phosphate, so no energy consumption there, and it is reducing NAD plus into NADH. So the molecule itself was oxidized, and you can see that because an aldehyde was converted into carboxylic acid, and in organic chemistry that's called oxidation, and then you get NADH. All right. The next step is 3-phosphoglycerate kinase. Recall this was the first ATP generating step, and the idea here is you transfer one of the phosphates on 1,3-BPG to ADP, generating ATP, and we call this substrate level phosphorylation. All that means is that you are phosphorylating without oxygen present. So we'll see when we talk about the electron transport chain in the next slide deck that over there you have oxidative phosphorylation when oxygen is present. Here you have substrate level phosphorylation. Next, we have pyruvate kinase, which is the last step of glycolysis, where you generate the next ATP molecule by transferring a phosphate from phosphoenolpyruvate to ADP, and this is also an irreversible step. And by the way, irreversible steps will show up again when we talk about gluconeogenesis. And finally, this pyruvate kinase enzyme is stimulated by fructose 1,6-bisphosphate. So what does that mean? Take a look at it's this molecule over here, fructose 1,6-bisphosphate, which is the outcome of the rate-limiting enzyme, actually activates this very last step, and that's what we call feed-forward activation. All right, now let's talk about key intermediates. Dihydroxyacetone phosphate, that 3-carbon molecule that we saw generated over here, can be involved in fat storage because it can be converted to glyceraldehyde 3-phosphate and then subsequently glycerol, which is the backbone of triacylglycerols. And then 1,3-BPG is also important because in red blood cells, in erythrocytes, it can be converted to 2,3-BPG, and then it can impact the oxygen hemoglobin dissociation curve. Again, I talk at length about the oxygen dissociation curve in a separate video in this playlist, but for the purposes of this video, all you want to know is that 2,3-BPG will shift it towards the right, so to make it have less affinity to oxygen. All right, now going back to our first diagram over here, you can see that glucose, after it's converted to pyruvate, can have one of 2 fates. If oxygen is present, it'll go down the aerobic metabolism route with the Krebs cycle and the electron transport chain. If oxygen is not present, then you will have anaerobic fermentation, and that is what we're going to talk about over here. Now, it turns out, like if glycolysis were to run indefinitely, you will run out of NAD+. So at this step, recall that you need NAD+ to convert glyceraldehyde 3-phosphate to 1,3-BPG. In this process, you generate NADH, and at some point you may run out of NAD+, and at that point, glycolysis can run no more. It turns out that our body, that our cells have this enzyme called lactate dehydrogenase to regenerate NAD+. So what it does is it takes NADH and it oxidizes it and reduces pyruvate to generate lactate and NAD+, so that glycolysis can continue to run. Now, importantly, if this process happens too much, where you're relying exclusively on anaerobic metabolism, you can have a condition called lactic acidosis. You don't have to know really the clinical details of this. Just know that if you have too much of this fermentation process occurring, you will develop too much lactate and then get lactic acidosis. In yeast, so yeast cells will have a different form of fermentation than us. They'll have pyruvate being converted to acetaldehyde, and then that converted to ethanol plus CO2 plus NAD+. So again, they're regenerating the NAD+, but just in a different way. All right, next let's talk about the pyruvate dehydrogenase complex, and this is that other route, like if you had oxygen present after the process of glycolysis, this is the route that will lead you to the Krebs cycle. Okay, so you can see the bridge between glycolysis and the Krebs cycle, and essentially what's happening is you're taking that pyruvate plus NAD plus and coenzyme A, using pyruvate dehydrogenase complex to generate acetyl-CoA, and then acetyl-CoA can go into the Krebs cycle or fatty acid synthesis. More on the pyruvate dehydrogenase complex in the next slide deck in part 2 of carbohydrate metabolism, which introduces the idea of aerobic respiration. I thought discussing the PDC fits better over there since it is indeed the first step of aerobic metabolism. All right, now let's shift gears and talk about glycogenesis. So glycogenesis occurs in the cytoplasm. It occurs primarily in the liver and skeletal muscle. So that's glycogen synthesis will occur in the liver and skeletal muscle. It's triggered by insulin, so you want to think about insulin being released in the fed state. So after you've had a meal, insulin will be released and you'll start storing away glucose because you have an abundance of it. And then it's inhibited by glucagon and epinephrine because those want to dump glucose into the bloodstream, which we don't want to do after a meal. All right, so key steps. First of all, you want to activate glucose to be able to synthesize glycogen. To activate glucose, you're going to use these enzymes, not extremely high yield. That first enzyme, you probably recognize it from what we just saw with glycolysis, hexokinase phosphorylates it. Then you use a mutase to change it into G1P. And then you use UDP, glucopyrophosphorylase, to generate UDP glucose. And that is the activated glucose that can then feed into the growing glycogen chain. And then if you recall from our video on carbohydrate structure and function, glycogen is not only a linear chain, but it also has branches. But the process starts by building the linear chain. So by joining the glucose molecules at alpha 1,4 glycosidic bonds. Again, this is high yield, but we discuss it in our carbohydrate structure and function video. So you start to add them using an enzyme called glycogen synthase. And by the way, this is the rate-limiting enzyme of the process of glycogen synthesis. Now, next, you want to start branching out. So the way that this branching enzyme works is that it will break a bond containing like 3 to 4 glucose residues, and then it will transfer them and branch out. And importantly, that branch will come out at an alpha 1,6 glycosidic bond. So essentially, these are attached by 1,4 glycosidic bonds. This sugar molecule and this one are attached by glucose 1,6 glycosidic bonds, and then the remainder are going to be attached by glucose 1,4. So it's really just the first one that's branching off. Okay, now let's talk about glycogenolysis. So glycogenolysis is the process of breaking down glycogen into separate glucose residues so that you can use them in the fasting state. So this is when you're fasting, you need access to glucose. Obviously, you're not getting it from food because you're fasting. So it's going to be triggered by glucagon and epinephrine, and it makes sense that when you are trying to dump glucose into the bloodstream, you have multiple hormones allowing you to do that. Because what happens if you're a gazelle being chased by a lion and you need glucose right then and there? You don't want to rely on only one hormone. You have multiple hormones at play over here. Now, glycogen breakdown. Glycogen is stored, as we mentioned earlier, in the liver and in muscle cells. However, it can only be released as individual glucose residues from the liver into the bloodstream. The muscle cells cannot release glucose into the bloodstream, and we'll see why when we discuss the enzymes in just a bit. So you can see what's going on over here. In order to cleave the linear chains of glycogen, you're going to use an enzyme called glycogen phosphorylase, which phosphorylates one of the glucose molecules and releases it. Now, importantly, we're not using ATP over here. We're using inorganic phosphate, so we're not wasting energy over there. Okay. Now, after that, you convert it from glucose 1-phosphate to glucose 6-phosphate, and then keep that in the back of your head. Let's talk about how debranching enzyme works. It works essentially the reverse of branching enzyme. It's going to debranch. So it's going to, let's say you had 7 glucose residues over here. It's going to take off 3, attach them here, take off another 3, attach them over here. And then this very last one is going to come and break that alpha 1,6 glycosidic bond. So essentially it's working as a transferase where it transfers glucose residues, and then it also has alpha 1 glycosidase activity. Okay, now the reason why muscle cells cannot release glucose into the bloodstream from glycogen is because they lack the final enzyme responsible for the breakdown of glycogen, which is glucose 6-phosphatase. So recall over here, we mentioned that you have glucose 6, the final molecule here is glucose 6-phosphatase. This cannot cross the plasma membrane, so you need to remove that phosphate group before it can cross the plasma membrane, exit the cell, and enter the bloodstream. Okay, and because muscle cells lack that enzyme, glucose 6-phosphatase, they can only use the glycogen that they break down into glucose inside of the cell. It cannot be released. And that makes sense. You know, muscle cells, they need the glucose, so they're not going to release it into the bloodstream, whereas the liver can release it into the bloodstream. Okay, now let's shift gears and talk about gluconeogenesis. So gluconeogenesis is the process of building up glucose, so creating glucose from pre-existing precursors such as lactate, oxaloacetate, glycerol, and glucogenic amino acids. And glucogenic amino acids, by the way, include all of the amino acids except lysine and leucine. So the idea here is that you want to reverse glycolysis, but glycolysis has certain irreversible steps, notably 3 irreversible steps. The very first one, hexokinase, the rate-limiting step, PFK1, or phosphofructose kinase 1, and the very last step, pyruvate kinase. So essentially, we're going to want to reverse these, and the way you reverse them is that you use other enzymes to bypass the enzymes that are irreversible. So for that, so we're going to start from the bottom. So you start with pyruvate, and it turns out you can't just go from pyruvate to the one right before it, which recall was PEP. You can't just go in one step over here. So that pyruvate kinase, we need 2 enzymes to be able to reverse it, and these are pyruvate carboxylase, converting pyruvate to oxaloacetate, which we'll talk more about when we talk about the intermediates of the Krebs cycle. And then the next step is to go from oxaloacetate to PEP using the enzyme PepCK. So these 2 enzymes will reverse the activity of pyruvate kinase. Next, the next irreversible enzyme is PFK1. And remember what the outcome of PFK1 was? Well, it was fructose 1,6-bisphosphate. So you need something to be able to go from fructose 1,6-bisphosphate to fructose 6-phosphate. So you want to dephosphorylate it, and you're going to use fructose 1,6-bisphosphatase to reverse that process. This bypasses PFK1, and it's inhibited by AMP, and that makes sense because when you have a lot of AMP, that implies that you don't have a lot of ATP. And the process of gluconeogenesis is energy expensive, so you don't want to use the little ATP you have, so you shut it down. Also, fructose 2,6-bisphosphate inhibits fructose 1,6-bisphosphatase. Recall, or actually we'll talk about this, I believe, we'll talk about this more in the next slide. Finally, glucose 6-phosphatase is what's going to reverse the activity of that first enzyme, which is hexokinase. So that's how you go from glucose 6-phosphatase to glucose 6-phosphate to glucose. And that's why, again, muscle cells cannot synthesize glucose and release it into the bloodstream because they lack that enzyme glucose 6-phosphatase. Let's talk about the regulation of gluconeogenesis. So it's stimulated by glucagon, cortisol, and epinephrine. Again, gluconeogenesis is a process of wanting to survive. So you want to rely on more than one hormone to be able to stimulate it, okay? And this is something I didn't mention earlier, but essentially glycogen is being used, like your glycogen stores are being used in the first 6 to 12 hours of fasting, whereas gluconeogenesis is really going to start being used once those glycogen deposits sort of start to deplete, so really like 12 to 24 hours later. Okay, so and it's also stimulated, it happens to be stimulated by acetyl-CoA as well. It's inhibited by insulin and AMP, and we mentioned why AMP would inhibit it, because then you have too little ATP, and you don't want to waste ATP to synthesize more glucose. Also, this concept of reciprocal regulation is important, and this is the idea behind fructose 2,6-bisphosphate. So recall, fructose 2,6-bisphosphate, we saw this earlier, stimulates PFK1, which is an enzyme of the glycolysis, so it stimulates glycolysis. Well, it turns out that fructose 2,6-bisphosphate not only stimulates glycolysis, but also inhibits gluconeogenesis, and that's the idea behind reciprocal regulation, is that you don't want 2 pathways that are accomplishing opposite things running at the same time. That would be futile. They're doing opposite things, so you don't want them running at the same time. So when fructose 2,6-bisphosphate is high, it's going to stimulate PFK1 and inhibit fructose 1,6-bisphosphatase, which is the enzyme accomplishing the opposite of that in gluconeogenesis. And when it's low, fructose 2,6-bisphosphatase can carry out its activity, and PFK1 will not. All right, this is a summary table. You can take a screenshot, pause the video, or just access our slide decks after the video. Also, a lot of these are covered in our Anki cards. And these are some knowledge checks. I'm gonna push through, but if you have any questions, don't hesitate to reach out to me and I can provide you with explanations and clarifications. All right, pause the video. Okay, and our last question over here. If you've found this video helpful and like the way I teach, I'd recommend watching the rest of the playlist. I spent a lot of time making sure that the videos build on one another, so it makes sense to watch them in order. Also, don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow and I would really appreciate it. Also, did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide decks. Finally, these videos, at least as of now, are sponsored by you. So if you're interested in supporting me to be able to continue to make these high quality MCAT playlists, you can support me either via Patreon or by buying me a coffee. Links in the description down below, as well as the links for everything else, including the link to our growing pre-med companion Discord community. Thanks for watching, and I'll see you in the next video.